I'm Bill Weiss, professor of dairy nutrition at Ohio State University.
I'm Alex Tebbe, dairy technical sales specialist for Purina Animal Nutrition, ex-graduate student of Dr. Weiss.
An ex-undergraduate student from the University of Illinois and dairy club president. Don't forget all your credentials. So we are here, and there is this article in press that Alex and Dr. Weiss co-authored, and the title is "The Effects of Oscillating Dietary Crude Protein Concentrations on Production, Nutrient Digestion, Plasma Metabolites, and Body Composition in Lactating Dairy Cows." And it has a lot of work in this. I can feel the weight of the paper, and some very unique measurements that maybe we don't see that often. Maybe we should see more often. Some classical measurements here that we can talk a little bit more later. But if you guys could maybe start by telling us how did you get to this idea? Where did you get this idea about, hey, I'm gonna oscillate this crude protein, and this is gonna be a good thing or a bad thing?
Phil, many years ago, probably 6-7 years ago, Norman St. Pierre worked at Ohio State then, and he and I were talking one day about why two herds that are fed about the same facilities, why can they differ so much in production? And one thing we thought about is maybe the higher producing herds just do things more consistently. The diet is consistent day to day. And, we've heard that forever, that, cows like consistency and so on and so forth, but there's absolutely zero research supporting that statement, zero. And so we set up a series of experiments where the basic design is we'd have a controlled diet that's a good diet and was as consistent day to day as we could make it. And then we'd have treatments that we varied. We varied the fat concentration. We varied protein, which we'll talk about today. We varied fiber. We just varied things. So we weren't looking at how the effect of concentration of protein, for example. What we looked at is if two diets are equal on average, but one is consistent and one isn't, does it matter? And that's where this started. And one of the first ones we did was a protein study. That was several years ago, and then we had some things we didn't really like, and so when Alex started here at his PhD, we wanted to repeat this but do it better, and that's the paper that Alex will be discussing here in details. But the theory was, what we started with is that consistency would be better. What we're finding in all this study in total is that it really didn't matter, or inconsistency might actually be a little better. So, but I'll let Alex discuss the details of the experiment.
Okay, so Alex, how did you guys come up with the treatments assortment? So you have 109 MP, right? And
correct.
Then you have a 95 MP, and then you have one that is oscillating. So pretty much you have two diets, and then you're kind of having three treatments out of those two diets, right?
Correct. Yeah. So the idea is can we use this oscillating method, so feeding a different concentration over a period of time, and then switching that concentration again for a series of time. We thought that would improve nitrogen utilization efficiency if we were feeding a diet that was marginal to begin with. And so that's why we chose those levels. We wanted an adequate diet that we could use as our control. And then the theory is if you reduce the MP to slightly marginal, that you'll add some efficiency when you oscillate the crude protein concentration day to day, and then that would improve the overall diet, and those cows would produce more milk than if you would just feed that same deficient concentration continuously.
I see. So it was an idea of exploring the nature of the ruminants to kind of, hey, let's have this cow recycle a little bit more. And did that start actually, and I have this question by the end, but I'm going to put it right here. Did it started like with the economics saying, hey, if I go from 16 to 14% crude protein, I may be saving X amount of dollars. So if I do the oscillation, actually, you should be saving money, isn't it?
That's correct, Phil, too. We all know protein's an expensive nutrient. So if we can get away with feeding less protein, but still have, and also increase production, you know, there's multiple other benefits, including reducing nitrogen excretion too. That's overall good for economics and the environment.
I see. You didn't end up putting anything on a table or something. Like what would be the cost associated with one or another treatment?
So it does decrease production cost because soybean meal that we replace with corn and soy hulls is a lot more expensive. So it was a cheaper diet as well.
Easy to cut diet. You reduce protein in a diet, you almost always reduce cost, but you almost always reduce milk production too, which means it's less profitable. So the theory here was we reduce cost by feeding a lower protein diet, and I can calculate what that would be, but it was substantial. But then by oscillating, you don't lose the production. So you'd save money on diet, but not lose production, which then would make it a profitable thing. Just reducing feed cost doesn't mean it's profitable if you lose milk. So
I see, I got it. And then you went from, in this paper, in a 24 hours, so one day was a high crude protein, the other day was a low crude protein. And so on average was equal to the 95 MP, but there's some buildup here that actually you did that using 48 hours before, and then you decided, no, now I'm going to do 24. I think that's the right amount. What was that?
So originally this research kind of took off in growing ruminants and sheep and cattle. And what they found was 48 hours was basically an optimal time to oscillate for growing ruminants. But when we tried this a number of years ago, a master's student of Bill's had basically took the research straight from what they had found worked well in beef and sheep, tried to apply it in dairy cows, and it didn't exactly fit. And when we got to thinking about it and looked at this deeper, what we found is that our dairy cows, they lag about a day in what they eat, versus growing ruminants, it's probably closer to 2 days. And it makes sense when we think about how much more feed dairy cows are eating. I mean, that's how much faster the rumen turnover time probably is in a dairy cow versus a growing ruminant. And so to prove that, we did a little pilot study before we actually conducted this study. We fed different average levels of protein, and then we also changed the oscillation frequency. So we tested oscillating every 48 hours versus every 24 hours. And we basically confirmed in that little pilot study that 24 hours is in fact better for dairy cows. But also as part of that is the level of deficiency that you're oscillating also can affect production. So then from that pilot study, we basically came up with these treatments and then tested them in a study that we could more effectively say, yes, you know, in a commercial setting, this will work. And then we also, of course, evaluated other things like the digestibility, amino acids in plasma, as well as body composition to fully understand, how the oscillating crude protein method works, or to unveil, more details of if it is an applicable thing on farms or not.
So at that point, you were at in your pilot, you were at 95 as well, or you tried lower, or?
So we tried 2 levels. The first level was 95, and then we also had tried, I think it was like 85% of the MP requirement. And when we oscillated 85% of the MP requirement, those cows did end up reducing their production. It was just too low of a concentration to maintain adequate rumen fermentation and amino acid supply, basically, for milk production.
I see. So the idea of reducing and oscillating and doing well means not losing milk production. That was pretty much a big goal, right?
Correct. Yeah.
And so that's one of the things that you guys conclude and you achieved, that, hey, this cow's the milk yield was the same. However, you saw some difference in intake, right?
Yeah. So compared to continuously feeding 95% MP versus oscillating, there was no difference in milk production, but those cows that were on the oscillating diet did tend to have less intake. And so we felt that, that might have been the limiting factor of why our hypothesis basically was incorrect, that it doesn't improve milk production.
But then that doesn't mean that you increased efficiency, or no? Because those cows, they produced the same amount of milk and they ate less. So that means they were more efficient, right?
Correct. They would have been more efficient whenever you think of milk divided by dry matter intake, but the nitrogen utilization efficiency, whenever we calculated that, which was one of our main objectives, was not improved with this oscillating crude protein.
I think it's important that you highlighted, is that if you're talking about feed conversion, pretty much, right? That could be a positive thing. But so you guys were dealing with mid-lactation cows, so around 119 days in milk, something like that. And then you fed that strategy for 50 days, right? So it's kind of also interesting to think about it in other stages of lactation, how that work, or if you would do that. Do you think that would be a good idea to do for fresh cows or cows at peak, or?
Fresh cows, I think it would be a catastrophe. I just think it would be an absolute catastrophe because they are changing so much day to day anyway, and they're in a very significant protein deficiency most of the time. So I don't think this has any applicability to that. But after that, once intake gets up, then I think it worked fine. It may actually be better in later lactation because, again, efficiency is going to make the money there. You're not going to get more milk at later lactation. But we haven't done it, but I really can't see any use in a fresh cow with this approach.
Based off of some of the other research that we've done, and Bill mentioned, fresh cows need probably even more MP than our mid-lactation cows, just because they're eating less. And so if we oscillate from a high to a low concentration, that would probably be a catastrophe.
This paper here and this research kind of touch a little bit about, I think, where Beefcatter and feedlot, they do a concept sometimes that they not only feed for a sleek bunk, but they play around about restriction as well and try to get that compensatory gain throughout the system. And for some reason, we don't do that in dairy, heifers or any stage of, so it's kind of, you are playing with crude protein, but the same thing you could play with, instead of feeding ad libitum, you restrict those cows for a certain amount. Let's say you feed 95% what they need, but then you're going to cut everything, not just crude protein,
Mm-hmm.
right? And then the next day you give 100%, and then the other day you feed 95. Do you guys have experience with that? Not only crude protein, but the whole diet. I'm just going to oscillate that.
One experiment we did kind of is like you said, not exactly, but where we basically added a lot of water to the diet, so we changed things. Cows did better. And again, that's why we're finding these inconsistent diets actually do equal or a little bit better. We think that had a lot to do. We didn't measure. Well, yes, we did. I forgot we did measure digestibility there. On an energy basis, these again were mid-lactation cows. So on an energy basis, those cows would have been more efficient, where we went from extra energy to less energy with carbohydrates, not fat. So I think there's some potential here, but a big difference between a lactating cow and a growing cow is, if you lose milk production today, you don't gain it back tomorrow. Whereas with growth, if they grow a little slower today, they can grow faster tomorrow. And lactation doesn't work that way. So we have to be careful on stunting them too much on production, because again, it's a different physiological thing than growth. So,
no, that's a very good point. Yeah. And then, so then you guys oscillated the crude protein, same milk, and then you had a lower intake. And then one of the things that you guys, again, we are talking about just measuring intake and milk, but you guys also measure the whole nitrogen. So a subgroup, you guys, of those cows, you guys put those into some collection chambers where you're able to do total collection of feces and urine. Is that right?
Yeah, so at OERDC and our research farm, we have the capability to separate the urine from the feces and do total collection of each on the cows. So basically we can calculate total balance of nutrients. And so we did that for crude protein and some of the carbohydrate fractions as well.
And then you guys have put straps on it to collect the urine. How does it work? Or you have the grades below, or you just
have a grade
yeah.
student with a bucket out there.
It's just like any fair that you go.
Well, we have a system. I can send you a picture if you want to include it. It's
yeah, that'd be cool.
We have harnesses that we have these what we call urine collection devices that strap onto the back of the cow, and the urine goes down a tube into a container, and the feces just drop through a grate into another container. But I'll send you a picture of it.
Okay, that's cool. And then you just collect that, and then you guys did that for 4 days, right? Is that right?
Yeah. And we've done that in the 30 years I've been here. I probably have done that on 500 or 600 cows. So we're very good at it. And 4 days actually was when long ago we looked at how long we need to do it, because the shorter, these are very labor intensive measurements. So short is better. And 4 days was it was the most precise, and it was as accurate as anything else. So that's why we came to 4 days.
So you collect the 4 days, and then you mix everything for one sample representative of those 4 days?
Yeah, we'd sample every day, and then mix the 4 days into one composite sample.
How did you compare that to some of strategies, for example, that you don't have the ability to do the straps and everything else, where you do 3 or 4 days and then you collect every 2 hours or 4 hours, and then you kind of try to? We got a new guy here, Dr. Lee, and he's kind of looked at some of these urine markers, sampling every so many hours and compared to a total collection. We've done that as well, and what we've done is actually collect the total urine and then take a sample of that
measure creatinine and then see if we can do it. And on average, it's reasonable, but there's going to be cows where it's just terrible, just not even close.
So some individual variation.
There's individual variation, and and so if you did it on 15, 20 cows, you're probably going to get the right average number. But if you're just doing it on a few cows, you have to be very careful of that. So it's to me, if urine nitrogen or urine energy, whatever, is a primary measurement, I'd be leery to use markers as my only measurement.
If he could explore a little bit on, what are easier assessment, because probably then we could have more done in other trials, right? Maybe with more cows, but less precision, I don't know.
Yeah. Well, Alex got another paper on, actually it's on magnesium, but he looked at this estimating urine magnesium with total collection or with markers. And he can go into that. I can't remember everything he found, and then this Dr. Lee has done some of that with nitrogen as well. So some people have looked at this as ways. And again, precision is poor. Accuracy with enough cows looks to be reasonable.
So whenever we compared it to total collection using creatinine versus actually measuring volume of urine produced each average across those 4 days, on average it was accurate. But as Bill had mentioned, there's a lot of variation from cow to cow, so you would probably need more animals. Some of the stuff that Chan-He was trying to address was on the optimal number of times that you need to sample throughout the day to capture the diurnal variation in creatinine production. And so I think he would be a better resource. I forget what they had found was the optimal amount of times that you would need to spot sample to capture all that diurnal variation. But yeah, they've looked at that as well in fecal markers to measure digestibility.
And then coming back to this trial about oscillating crude protein, and then something that, I found it very interesting is that how cows manage to switch where they are excreting nitrogen. It's kind of, I found it interesting where they kind of move from urine to feces. Were you guys expecting that, or?
So whenever I got the results and started to look into it more, there is some studies that have shown that you increase crude protein digestibility, but they didn't find a subsequent increase in urine nitrogen. And so our thoughts is we switched the route that the urea was being recycled from the large intestine to basically going more towards the rumen. But in our study versus the other studies, why we saw an increase in nitrogen than in the urine, because that greater amount of urea that was being recycled to the rumen was not well utilized to make microbial crude protein. And so that might be the partial reason why we switched nitrogen excretion from feces to the urine.
And one other, he saw, Alex found a pretty significant increase in nitrogen digestibility, which is a good thing if that animal then uses that nitrogen to make milk protein. And they didn't in this case. They digested it, but then they oxidized it and excreted the nitrogen. So that would suggest that energy, milk protein responds quite well to increased energy. So this would kind of suggest maybe energy was involved here too. And we didn't, we need to work on getting the energy. If we're going to increase the digestibility of the nitrogen, we got to make sure the energy is there to use that nitrogen to make milk protein. So one thing, if we continue this line, is how can we then increase energy supply along with this oscillation. And that's this drop in intake. That's the problem, is we lost energy intake as well, and that they need energy to make milk protein.
Right about that, about, hey, you know, we need to explore this a little bit more. And I agree. And even if we do, you know, like high starch, low starch, or you are manipulating, you kind of get confounding on some things. So when you are manipulating your crude protein, you are changing some energy sources as well. And that's kind of what you can do, right? So I think you start answering one question at a time, and now you start, oh, maybe I can. So do you think that, well, first, I know that's kind of not, how can I say, ideal to do it. But for example, if you get your results from nitrogen and purine derivatives, and you try to play around, hey, how much microbial protein I would have based on this energy sources that I have, would that show big differences as well, or no? I'm trying to assess is, for example, if you think if you had a different source of energy or the way you played with the ingredients, that would give you the next step, you think?
Yeah, so I think that would be a very interesting follow-up to this is, you know, like you mentioned, when we reduce crude protein, something has to go back into the diet. Whenever we set up this study, we chose to add in starch and NDF and fiber back into the diet, and the reason for that is if we add more fiber, we may lose intake, but if we add all starch, we may cause milk fat depression. So we thought, well, let's do 50-50 replacement, because that would be, you know, the most optimal in for what we're trying to accomplish. But that would be an interesting follow-up to see the effect of oscillating crude protein and starch, basically, or crude protein and fiber, versus a 50-50 mix.
But let's say if you add molasses or other sources of energy that not necessarily are just starch, so you kind of get away from, or beet pulp or whatever, that you get away from your milk fat depression, but you're still kind of balancing that energy. But then I'm not sure how, if the software will capture that, if the NRC will understand all those fractions and say, oh yeah, this is a good match here. You have all your protein profile, your ABCs here. Now I'm doing this with soluble fiber and sugars, you know. I'm not sure how sensitive that system is to tell you, yeah, this is how you should go, right?
I agree with, you know, I don't know what the software out there right now, I think would do exactly opposite of what we want. Because, you know, when we drop the protein and the days where we fed low protein, it's going to say, well, you don't need energy to capture the nitrogen because it's not there. So it's going to say feed less energy. And one of the big problems is we're so tied to a day, and we forget that this cow, what I feed today is going to affect this cow tomorrow. It's not just going to affect it today. And we're off this cycle. And you know, if you look at Alex's MUNs, it's one day off. The day of high protein had actually the lowest MUNs.
Yeah, that was very interesting. You think the opposite,
so,
right? It's high should be low, you know.
And it's so we just got to quit thinking everything's a 24-hour cycle. It's not. The rumen isn't totally empty when we feed it the next day. You know, it's still a lot of stuff left over from the day before. So the software we have right now on a day, if you do it on a day, wouldn't work. We need to move it ahead a day. But I think of coming up with ways. Sugar's a good idea. You know, it's not a big milk fat depressor. It helps on energy. It's highly digestible. So that might be a good idea. But you know, his diets, his high and low protein differed by about 5 percentage units in protein. So you have to make that up with something. 5 units of starch worried us changing. And so that's why we went half and half. To be honest, we never thought of sugars. That might have been a good idea to get some energy in there as well. But what you're substituting, I think, is an area that we really need to think about.
Through and analyze a lot of amino acids, right? And then all that relationship of energy and amino acids, or let's say energy and protein, I don't think there is much in-depth understanding of where that energy is coming from. If it's from fat, from carbohydrates, maybe that's what we are more used to, or sugars, and what types of sugar. I think it's a pretty cool area as well. You think that going backwards, so see what your cows did with the purine derivatives and everything else, that matches what the software was telling you, or no? So if you go to say, hey, my cows, they are producing this much MP, or that's the prediction, and does that happen? Or because I think what you said about, hey, this cow is not empty and we are feeding a day, I never thought of that, but that's pretty important, and I'm not sure how to fix that.
If like I said, I think a lot of this was, at the end of the day, the reason we didn't see the maximum benefit of oscillation is energy intake. I'm just, however you're going to, whatever form of energy is the reason. So again, and it's not so, we measured digestibility. It was about the same. So the DEs, digestible energy, is probably about the same. Methane, we don't know what that would do with oscillation, but that's not a major energy loss. We don't know what efficiency does, but metabolic efficiency. But it's still, to me, the thing we need to work on is to avoid that loss of 1 kilo of intake. That's a lot of energy. And so if we could come up with, and we don't know why that went down, we don't know why, but if we could figure out a way to prevent that drop in intake from oscillation, that I think would fix a lot of things. These cows would have done what we hypothesized that they would do.
Mm-hmm. And why, just curious about your 109 treatment, was that just to prove the concept that the 95 was different than 109? Because it seems like, that oscillation was really your objective or your hypothesis. And so was it 109 there just to prove that the 95 was different and say, hey, I have a good control here, like positive, negative type of deal?
Yeah, that in the first experiment Alex was talking about, but we didn't have a good negative control, because if we had just the 95 and the oscillation, and let's say we got the same production, which about what we got, that doesn't mean it's as good as more protein. It doesn't prove a thing. Mm-hmm. So we had to have, if Alex didn't see a reduction in milk with the 95, we couldn't have made any conclusions at all, because we didn't challenge the subject, the cows. But he got less milk with the low protein, and then the theory would be we'd gain that milk back from oscillation. So we needed a good negative and a good positive control.
No, that was good. And then, Alex, tell me more about this body composition thing, because I think you got something pretty cool out of there. Like when you're oscillating, is the cow making that up from where? Where is that coming from? If that would be from muscle or for somebody else, but that seems pretty labor intensive.
Yeah, so it was, so basically what we did and how the theory works is we injected urea into the cow's blood, and that urea will equilibrate with all the body water. And so because body water and the amount of protein is pretty much constant, you can figure out how much body water, body protein, and body ash, and then by difference determine body lipid based off of how urea concentration changes before and after you inject that set amount of urea based on body weight. So then what we did is we used that urea dilution method before we actually started treatments, And then at the end of 50 days, to try to track, is there a change in the body composition? So is this what we had hypothesized? Is this enhanced milk production that these cows are having on the oscillating diet? Is that because they improve their efficiency, or are they actually mobilizing protein off their backs to spare the loss in protein that is coming from the diet? So we wanted to ensure and eliminate that as one of the factors improving efficiency when you oscillate.
But you have to do that. So you pretty much just infuse, and you don't even need to leave a catheter there. Then you go to the tail and collect blood samples as you go.
Right. So there was a large study done, I think it was at the RAU in Europe, where they evaluated body composition after doing the urea dilution method, and they found that worked well compared to actual total body composition measurements by measuring all the components and then measuring chemical composition. That if we infuse urea into the jugular vein and then 12 minutes later take another blood sample out of the tail vein, that you could determine what we call the urea space volume of those cows, or the amount of body water basically can be calculated.
And then do you remember what you found in this paper?
So overall, we found that cows, they didn't change their protein, total body protein. So whenever they received less protein in the diet, they just reduced their milk production. They didn't change any of their body mobilization. But we did find that they were in positive energy balance, because over time they gained lipid. And then overall, there was no treatment effects on body composition. So whether you fed high or low protein or oscillated, total body lipid and protein was unaffected.
I see. And you also measure plasma 3-methylhistidine, and kind of confirm the same story, right?
Correct. So with 3-methylhistidine, it's a biomarker for skeletal muscle catabolism. And so if you have an increase in 3-methylhistidine, that generally means you're having mobilization from skeletal muscle. And we didn't find any differences in concentration across treatments to basically confirm that our body composition methods using urea dilution are correct.
Do you see then mobilization if you would do 48-hour oscillation, or no? It's same story.
So we haven't evaluated it in the 48-hour or that pilot study. We just evaluated it in the research that we published in this paper, oscillating every 24 hours. I think you would find the same results, that cows, they'll just reduce their milk production when they're in mid-lactation. They're not going to mobilize muscle if you oscillate just for 48 hours.
When you guys did that for 50 days, did you guys do it before for 40 days or 30 days or 60? And you say, no, 50 gives us a good idea. You think that more time, probably if the direction, it would be the same? If you left those cows 100 days in your trial, you think body composition would be the same thing, right?
I think so. And that's based on some other studies looking at protein in general. At this stage in lactation, you get up around 7-8 weeks, it's an accurate response. So, we thought 50 days is a good compromise between cost, experimental cost and accuracy. So,
no, I think it's I think, like I said, it's a pretty neat research. I think we learn about, nitrogen. We learn about this oscillation, just, because sometimes I think just because we feed the same way all the time doesn't mean that needs to be done, right? I mean, one thing, how was the the idea of feeding those diets one day off, one day, that was just feeding two diets to different cows, right? Did that give a lot of management problematic for you to feed those diets, or no? It was fairly easy.
So that's a question that always comes up is, you know, consistency is better for workers, but I think today with our software that we have available, and the logistics behind those softwares, when people are using or are mixing feed for cows, they're basically confined to what's next on the ingredient list. So it worked really easily that we just changed the diets in the computer every single day. And then our feeders, whenever they're feeding the cows, you know, the ingredients pop up that they need to add in, and there's no sort of question or wait a second, you know, did we feed high or did we feed low yesterday? So those type of questions didn't come in because we are basically controlling that by the software.
Do you guys have any final thoughts or considerations? You've been very generous of your time from Ohio or from Illinois.
Well, I guess, you know, a couple of things here. One is, you know, we need to be careful the way we feed cows, but cows can take some, we don't have to be perfect every minute of the day. This oscillation as a way to improve nitrogen efficiency has to be, I hope somebody carries this on and again looks at ways to reduce this negative intake. Then I really think there's potential here. For both, you know, I just did a quick calculation based on feed costs in Ohio. If you get the same milk from these, you'd save about 20-25 cents a day per cow. So it's not a huge amount, but it's not a trivial amount either.
Yeah.
It helps on the environment, which we all have to be more concerned about. So there's a lot of potential here. We're not quite ready for prime time yet, but there's a lot of potential here.
Alex, final thoughts? Are you going to start applying that now as a new nutritionist at Purina? Do you feel confident of applying this technology in your farms now or not yet?
Well, like Bill had mentioned, I think we're not quite ready for prime time. Maybe there's one or two more studies that need to be conducted to, you know, figure out what's the optimal switch in the protein that we're substituting. You know, is it starch? Is it sugar? Like we talked about. But also, too, you know, we often forget that protein is not all the same either. We have protein that's highly ruminal, degradable, and we have proteins that are high in bypass protein. So understanding what type of protein should we take out and what type needs to stay in to maintain intake or rumen microbial synthesis as well. So I don't think it's quite ready yet for prime time, but like Bill had mentioned, I think if somebody would pick up this research, we have some potential for it being commercially applied in the field someday. I also would say too, from this study is, and what Bill has shown is that cows are in fact very flexible. You know, that the classical, we need to feed the same thing day in, day out may or may not be true. And so understanding how we can play with the cow's rumen and that physiology to better optimize how we produce milk would be, I think, an important aspect for future research.
Yeah, that's a good point that you guys mentioned that I forgot about the quality of the protein, or maybe a better characterization of that protein, more towards microbial protein or not, that could impact as well.
For simplicity, all we changed was soybean meal. That was it. And so, you know, the rumen degradable protein, when we, on those low days, got to be pretty low. So maybe, you know, if we would have done something a little more complicated where we still change crude protein, but maybe we don't change RDP as much or things like that. So that's another area that could be or should be investigated.
Alex, now Purina can fund a lot of research at Worcester, Ohio. You're going to be there so you can keep doing the collections. When they say somebody keep up, that could be you as well, at least on the
That's right.
supporting, financial and labor intensive work. That's all good.
He should do that for his alma mater.
Exactly. Yeah. And you can still come back to Illinois for an official. I'm not sure if Ohio has an official, but we do. Okay, guys. Thank you very much.
Thank you.